Hi friends, welcome to our channel Way to Success. இந்த வீடியோவில் PGTRB Zoology Unit 1 Comparative Anatomy of Vertebrate Brain கான short notes பார்க்கலாம். Introduction. The brain is the main part of central nervous system. Brain is enclosed in the cranial cavity. In all vertebrates, the brain develops from the embryonic ectoderm. The early developmental pattern is very similar in all vertebrates. In all vertebrates, the brain consists of three main divisions. First one, prosencephalon or forebrain. Second one, mesencephalon or midbrain. Third one, rhombencephalon or hindbrain. The cavity present inside the brain is called ventricles. Development of brain. The anterior end of embryonic ectodermal neural tube is thickened, known as encephalon or embryonic brain. Encephalon undergoes differential growth and two constrictions appear, dividing it into a linear series of three primary cerebral vesicles. They are prosencephalon, mesencephalon and rhombencephalon. The first one, prosencephalon. Prosencephalon is divided into anterior telencephalon and posterior diencephalon. Telencephalon grows and forms paired cerebral hemispheres. From anteroventral part of telencephalon, paired olfactory lobes grow anteriorly. Cerebral hemispheres encloses lateral ventricles, first and second ventricles. Diencephalon has upper epithalamus, middle thalamus and hypothalamus. Dorsally, diencephalon gives out unpaired parietal body and pineal body. Ventrally, hypothalamus forms infundibulum that fuses with pituitary gland. Next, second one, mesencephalon. Mesencephalon has thick roof forming two optic lobes, that is 4 lobes in mammals, and two longitudinal bands of nerve fibers called cerebral peduncles or crura cerebri. Third one, rhombencephalon. Anterior dorsal parts form metencephalon or cerebellum. Remaining part forms myelencephalon or medulla oblongata. In higher vertebrates, the floor of metencephalon thickens due to the tracts of many nerve fibers and forms distinct pons varoli. Next, we can see about the ventricles of the brain. Cavities present in the brain is called ventricles. There are 4 ventricles. First and second lateral ventricles present inside the cerebrum. Third ventricle present in the diencephalon. Fourth ventricle present in hindbrain. The third ventricle is connected with the first and second ventricle by 2 openings called the foramen of Monro. Third ventricle communicates with the fourth ventricle by a narrow canal present in midbrain is called iter or aqueduct of Sylvius. Comparative account of vertebrate brain. First we can see for cyclostomes. Brain is very primitive without well marked subdivisions. Olfactory lobes are distinct. Cerebral hemispheres are small. Lateral ventricles are rudimentary. Parietal and pineal bodies are well developed. 2 optic lobes are imperfectly differentiated. Cerebellum is small. Transverse dorsal band. Medulla oblongata is very well developed with 6 pairs of cranial nerves. Fishes, elasmobranchs or contage thighs. Olfactory organs are enormous, so olfactory lobes of brain are proportionately large, attached to cerebrum by short olfactory peduncle. Cerebrum is undivided. Diencephalon is narrow with well-developed dorsal pineal body. Midbrain is very large with two oval optic lobes, center for nervous coordination. Cerebellum is especially large due to active swimming habit. To assist cerebellum in maintaining equilibrium, restiform bodies are present in the anterolateral angle of medulla oblongata. Next, teleus or ostrich thighs. Olfactory lobes are large without olfactory peduncles. Cerebral hemispheres and diencephalon are smaller. Optic lobes and cerebellum are larger than elasmobranch. The anterolateral sides of medulla oblongata shows unusual bulging or vagal lobes for taste. Next, amphibians. Forebrain is large. Olfactory lobes are smaller, but cerebral hemispheres are larger in accordance with more complex activities of locomotion, hibernation, breeding, etc. Cerebral hemispheres are separated by a median longitudinal fissure. Diencephalon is short with vestigial pineal body. Midbrain is small but well developed, forming two large optic lobes, corpora digemina. Optic lobes are the probable dominant coordinating center in amphibian brain. Cerebellum is poorly developed with just a transverse band on dorsal side. Medulla oblongata is short. Reptilians. Reptilian brain shows advancement in size and proportions over that of amphibians because of complete terrestrial mode of life. Cerebral hemispheres are larger, enclosing lateral ventricles. Two long olfactory lobes are connected to cerebral hemispheres, which are larger than that of amphibians because of greater thickness and the enlargement of corpora striata. Diencephalon is small and rounded. Midbrain has large optic lobes, may be the important coordinating center. Cerebellum is small, simple, and some pear-shaped, larger than that of amphibians. Birds. Brain of birds is relatively larger but short, broad, and rounded in shape. Olfactory bulbs are small and degenerate due to poor sense of smell. Cerebral hemispheres are very large and smooth, responsible for intelligent behavior and to control reflex behavior. Diencephalon is covered by cerebrum and cerebellum, bears a small pineal body. Midbrain is highly developed with large rounded optic lobes. Optic lobes are very well developed, correlated with keen sight and are laterally placed. Cerebellum is large with several superficial folds extending forwards. It is greatly enlarged due to many activities involving muscular coordination and equilibrium, such as flight and perching. Next, we can see about the brain of mammals. Brain is proportionately larger. Cerebral hemispheres are immense, projecting forward above olfactory lobes and behind above the diencephalon and midbrain. In Prototheria, cerebral hemispheres are small and smooth. In Metatheria, they are larger but smooth. In Eutheria, they are greatly enlarged with convoluted surface. The elevations of convolutions are called gyri and the depressions are called sulci. The convolutions are folding increases surface area of cerebral cortex, resulting in greater intelligence. Two cerebral hemispheres are separated by median longitudinal fissure and are joined internally by transverse band of fibers called corpus callosum. Olfactory lobes are relatively small but clearly defined and covered by the hemispheres. Diencephalon and midbrain are also completely covered by cerebral hemispheres. Characteristics of mammals are four solid optic lobes are called corpora quadrigemina on the roof of midbrain. The third ventricle or iter of midbrain is laterally compressed vertical passage called the cerebral aqueduct or aqueduct of Sylvius that connects the third ventricle with fourth ventricle. Cerebellum is also large, conspicuously folded and may overlie both midbrain and medulla. White matters look tree-like, called arbor vitae in grey matters. Hindbrain contains centers for the regulation of digestion, respiration and circulation. Thank you for watching this video.